Today I'll be sharing about one of my biggest scientific inspirations, Emmanuelle Charpentier, the microbiologist who co-discovered CRISPR-Cas9. I chose her because she reminds me that science is also about curiosity, creativity, and courage. Emmanuelle was born in 1968 in a town in France. From a young age, she was drawn to biology and chemistry, and her parents were extremely supportive of her academics. She studied at the University Pierre and Marie Curie, which is now Sorbonne University, where she earned her PhD in microbiology. Throughout her career, she moved between labs in France, Austria, Sweden, and Germany, often restarting her work from scratch, which is something that taught her independence and persistence. Outside the lab, she enjoys music, art, and long walks in nature. She's known for being quiet, reflective, and creative, which mirrors in the way she approaches her scientific problems. I admire Emmanuelle because she blends creativity and science, which are two things that I value. She has stayed true to herself while succeeding in a very male-dominated field. As someone in my family who is the first to study in the STEM field, I relate to her independence and the feeling of finding your own path through curiosity. Emmanuelle Charpentier is a microbiologist who studied how bacteria defend themselves from viruses. And while studying Streptococcus pyogenes, she discovered that two key RNA molecules, cRNA and tracer RNA, that work together with an enzyme called Cas9 to cut DNA. This was the turning point that led to her development of CRISPR-Cas9, which is a tool that can precisely edit genes. This discovery completely transformed the world of biology. CRISPR-Cas9 makes gene editing fast and accurate, which is something that once seemed impossible, and it's now used in medicine to fix genetic diseases, in agriculture to grow stronger crops, and in research to explore how genes control life. In 2012, she co-authored the paper, A Programmable Dual-RNA-Guided DNA Endonuclease in Adaptive Bacterial Immunity. The goal was to understand how Cas9 recognizes and cuts DNA. They combined purified Cas9, cRNA, tracer RNA, and a target DNA in test tubes. This was called in vitro assay. Using gel electrophoresis, they could see whether the DNA was cut. The results showed that Cas9 cut DNA only when both RNAs were present, and the cut occurred right next to a PAM site. Here's figure 1 from that paper. The gel image shows that the DNA cutting happens only when both cRNA and tracer RNA are added with Cas9. Without them, the DNA stays uncut. That's the top band in the gel, and the lower bands represent the fragments that were cut. This proved that Cas9 is a dual RNA-guided enzyme, meaning it needs both RNAs to make a precise cut, which is the foundation of the CRISPR gene editing. Emmanuelle's discovery gave scientists the ability to genetically edit many different organisms. Her story reminds me that curiosity, patience, and creativity can lead to discoveries that can change the world. She shows you don't have to be the loudest voice to make the biggest impact.